Okay, hello. My name is Arno Gutleb. I work in Luxembourg at the Luxembourg Institute of Science and Technology. And since several years, we work a lot on alveolar cell culture models at the air-liquid interface, which is also the topic of today's webinar. I want to start with a disclaimer. I do not have any economical interest. I'm not connected to any company that may show up during the presentation. And please consider this is not a systematic review. I only want to try to show some basic principles of the technology. If you think about why do we talk about in vitro assays at all, and probably you have seen this before, but there are huge costs related to it. Statements in 2005 consider costs of 10 billion euro for animal experiments worldwide. There was a use of 100 million animals. Also, on the other hand, many studies are required by current legislation, so companies may not even have a choice. The question is also of the value of these data. How can we transfer the results to the human? And that is very important when it comes to cell culture models for the lung, because there are big differences in the particle deposition between rodents and humans, which means that by definition, a rodent is not a very good model at all for human toxicology. In addition to that, there are differences in the immune system. In vivo studies very often apply very high doses, which means they are not realistic and not relevant. And because in animal experiments you want to use as much as possible all the information you have, you end up with large databases and you start to correlate exposure with a lot of endpoints, which of course increase the chance of false positive correlations. And very often we have to test compounds of low toxicity in animal experiments, which is by itself questionable. And I said, sometimes you are asked by risk assessors to provide this data, so you may not have a choice. That's why we try to develop and work on alternative in vitro test methods, especially for the lung. These assays should be based on human cells to be as close to the human in vivo situation as possible. We want to measure directly relevant endpoints. And another advantage is if you use at least air-liquid interface, that the dose determination, and we will come back to that later, is at the air-liquid interface quite easy. Why do we study air-liquid interface at all compared to classical cell culture where your cells would be covered by a cell culture medium? That's of course because in our breathing system, we have only lung. We are not exposed under cell culture medium. That would be the case of drowning. This slide shows you some of the differences why in vitro methods can be very valuable in pulmonary toxicology, and that's the anatomy of the lung. On the left side, you see the mouse, and because you have different diameters of all the various, let's call it simply pipes, all the way down to the alveoli, and you have a different anatomy of the alveoli, which is very short in a rodent, you get completely different particle depositions simply because the aerodynamics in the lung are different. And the second important aspect is simply the size. The size again implies that there are differences in airstreams, where the particle goes, where they deposit. So if you talk about models for the pulmonary system, you have a range of possibilities from relative simple models all the way to ex vivo models where you can use a complete lung. What I will talk today about is mainly 3D cell cultures at the air-liquid interface. Because you expose the lung cells to the surrounding air and you keep these cells not underwater, it's much more relevant. Again, we do not want to have cells under cell culture medium. Another aspect, and that's very important when you move from chemicals to nanoparticles, is that the very moment you put nanoparticles into cell culture medium, you will get the formation of what is called a protein corona, meaning that proteins will adhere to the surface of your nanoparticle, and dependent on what is at the surface, these are proteins or lipids that may be recognized by cells, altering the uptake so that the uptake rate is dependent on your cell culture medium. Now, if you keep in mind that at the air-liquid interface, your cells are exposed to the particles directly via the airstream, this is the method of choice for pulmonary toxicology. If you look into the literature, you will find a lot of cells have been used. You see skin, cornea, nasal, tracheal cells, bronchial, alveolar cells. Also melanocytes and even immune cells can be cultured at the air-liquid interface. You will find different species. A lot was done in human, but you find also papers on chicken and rat. And what can you test? This method was applied to particles, nanoparticles, virus, bacteria, chemicals, pharmaceutical, tobacco products, you name it. The number of chemicals is practically up to you that you can use. That's the big advantage, how you culture cells at the air-liquid interface. You keep them in something called the transwell insert, which allows you to expose them directly to particles, which is important, as I've said before. While in the submerged conditions, the particles will start to have interactions with the cell culture medium, and whatever reaches the cell may be completely changed compared to the air-liquid interface. Only one example that this has practical relevance. We don't have to go into the details of the slide, but you see on the left side air-liquid interface and on the right side submerged cell culture. These are cells exposed to the same zinc oxide nanoparticles, different endpoints, but with one blink of an eye you can see that the differences on the left and the right are quite clear. If it comes to human cell culture of the pulmonary system, you will find different cell lines have been used. You see airway epithelial cells, alveolar epithelial cells, 3D cultures, 3D human reconstructed airways. They are produced commercially by at least 2 companies, and there's even organ on a chip. Now you will see on my last slide the relevant references for these presentation and the slides. You will not find all of them here. If you cannot locate them, feel free to send me an email that you also will find on the last slide, and I'm more than happy to provide the PDFs of these papers. This is the anatomy of the pulmonary system. You start with the trachea and the bronchus, and you see there are many different cell types, tissues, from chondrocytes to muscle cells, you have glands, you have mucus. The cells in the airway have epithelium, they beat, they transport things out of the airway. The further down you come in your lung system, the more simple the system starts to be, the thinner the system starts to be, because that's a necessity for the exchange between oxygen and carbon dioxide from the blood side to the air side. And finally, in the aerosol, in the alveolar, you have a thin layer of only 2 cell types and the endothelium. And that's the system that we want now to mimic in the air-liquid interface culture. That is how it's currently done. On the right side, you see the anatomy again, the histology. You see a surfactant covering the 2 cells, epithelial type 1 and type 2, and the endothelial cells, meaning the blood capillaries. On the left side, you see how such a cell system can look in vitro. You have endothelial cells. On the lower part, you have the transwell inserts. You have epithelial cells on the upper part exposed to the airstream. And you can have different immune cells, like in this case, macrophages or mast cells. And as I said before, if you want to study particles, this is very important, even more important than for chemicals, because particles now can directly interact with the lung cells without any interference with cell culture medium compounds. If you have such a model, you have to, of course, consider that all the different elements, the 3-4 cell lines that you may use, do what they are supposed to do in vivo. On the lower left panel, you see immune staining for surfactant protein A and surfactant protein C. which means that the cells at the air-liquid interface have at least intracellular the important surfactant proteins. Now that means intracellular, you have to be sure that it's also extracellular, because this will allow the cells to survive at the air-liquid interface. Now, if you do this, there's a simple test where you have a blue dye of a certain surface tension and you put a drop on your cells. On the left side, you see epithelial cells grown at the air-liquid interface. You see a small spot, meaning there is a low surface tension. If you just take them out and they are grown under submerged conditions, you see the spot is already larger, meaning there's a difference in the surface tension. If you, for example, do the same assay on endothelial cells, you get a big blue spot. There is no surfactant present at all. The presence of this surfactant is an absolutely necessity that the cells can survive at the air-liquid interface. I said in the system there are mast cells. These are cells that produce and release histamine. In the lower left panel, you see in green immunostaining against histamine, meaning that our mast cells also produce histamine as they are supposed to do. Now we come to the macrophages. These are cells that are supposed to phagocytose particles that end up in the alveolar. And you see in green stained this cell. This is a CD11B staining, meaning this is a surface marker of mature macrophages, and they can freely move in this three-dimensional space in the transepithelial system. You see here in this animation one macrophage clearly marked, and within the macrophage you start to see particles. These are silica nanoparticles that were taken up by the macrophage, and you see that there are no other green whitish spot in the other cells, meaning the other cells also do not phagocytose as they are supposed to do. Now it is relevant, and I will show you one result. This is exposure of a multi-cell culture system containing 4 cells, and again have a look on the lower left panel. You see something like green cells with a black spot in the middle. This is a green staining for NRF2, which is a cytoplasmatic receptor that upon contact with benzpyrene and similar like compounds will go into the nucleus. As in the control cells, there is no compound present. The cytoplasmatic receptor is outside the nucleus. While if you expose it after 2 hours, 4 hours, you start to see agglomeration of the green dye, the green dye in the nucleus, in the cells. And this is now very interesting because these are endothelial cells, meaning you expose on the alveolar side, you have a contact with the epithelial cells, something is passing to the endothelial cells, and after 4 hours you see already a reaction on the endothelial side, and this could be the first onset of cardiovascular parameters. Now, already coming to some conclusions. If you build a system that you want to culture as the air-liquid interface, it should resemble the in vivo exposure conditions. Your cell system at the air-liquid interface will need the production of some protection. In the case of lung cells, this is the surfactant. We have seen that our cells produce surfactant. You saw one slide that if you start to test something, you see clearly immediately differences between ALI and the submerged culture systems. It's especially important if you start to study particles, because the particles will interact with the cell culture medium, which will result in a completely different exposure, and you do not study what will happen in the lung, in the airstream. And the dose is much better controllable in an ALI system than in cell culture medium. And overall, air-liquid interface is for the lung much more relevant than submerged conditions. I promised some key publications that are the ones that were taken and used in this presentation. You have my email address below, and feel free to contact me if you have questions. And with this and a Luxembourgish thank you, merci vielmals, I finish.